Good afternoon. My name is Jenna Blankenship, and we want to welcome you to today's EMSL Learn webinar, where we will be discussing upcoming proposal calls and capabilities that you can request for your environmental research. First, I want to share that we will be hosting 2 additional EMSL Learn webinars to help prepare you for the upcoming proposal call openings in December. On November 30th, we will be highlighting available capabilities in the biological research space. And then on December 7th, we will be highlighting call topics and available computing focused capabilities. More information will be provided through our social media channels and through our email newsletter about these upcoming opportunities. To follow along and make sure that you don't miss the details about these proposal calls, make sure you are subscribing to our email and social media channels if you haven't already. Megan Murray will be dropping the links in the chat. So today we have John Bargar, who will be introducing upcoming proposal calls and pertinent details in the environmental research or science research. Swarup China will be introducing the new terrestrial atmospheric processes integrated research platform, and he'll be discussing related capabilities and opportunities. Amir Ahkami will follow. He'll be discussing the new rhizosphere function integrated research platform and its associated resources. And finally, Dusan Velickovic will be talking about the novel MALDI MSI derivatization workflow for enhanced imaging and confident identification of primary and secondary metabolites. There's going to be time at the end of today's webinar for questions, so we encourage you to post them in the Zoom chat, and our speakers will answer them during the last 15 minutes. So with that, I'm going to toss it over to John.
Great, thanks very much, Genoa. So I'm John Bargar. I'm the science area lead for the Environmental Transformations and Interactions Group. And by way of introduction, first of all, happy Thanksgiving. Second of all, there are 4 different types of proposals that I'll be showing today. I just have 5 slides, 1 slide for the intro, basically, and 1 slide for each of the proposal types. And the other thing is that most of the user research that we fund every year is funded through these user proposal calls that we have that we release in the beginning of the winter, and then the proposals are due in the very late winter. So if you miss this opportunity, then you may need to wait a whole year. So for those of you who joined in today, you did absolutely the right thing. It was really good to focus on these calls. Let's see, so I need to, there it is, take control of the screen. And all right, so I guess I'll just go on to there it is, the next slide. So the first of the 4 proposal types is the so-called large-scale EMSL research program. This funds proposals, user proposals for up to 2 years. It advances scientific understanding in each of EMSL's 3 science areas, which I've highlighted here below in green, blue, and orange, environmental transformations interactions. That's what we're talking about today. And then functional and systems biology. So there will be, as Genoa said, there will be a future webinar for that, and also computing analytics and modeling, and that'll be covered in a similar future webinar. So we're going to be talking about environmental transformations interactions today. There will be 4 separate call topics that'll come out with a large-scale research call. They will, by and large, align with one of the 3 integrated research platforms that we have. And in today's talk, you'll hear Swarup China and Amir Ahkami introduce 2 of those 3, which actually are new. So this is a really cool opportunity to kind of get in at the beginning. So we also tend to highlight novel applications of EMSL capabilities. So the proposal call opens on December 14. Letters of intent are due on February 9. And yes, if you're wondering, a letter of intent is required in order to submit a full proposal. The deadline is March 23rd. I just want to emphasize two other things. First of all, the vast majority of these letter of intents are actually approved. So this is actually, and the second thing is that's because we have a very interactive proposal review and submission process. We use the LOI to understand what you want to do and provide really valuable guidance back to you before you go to the effort of writing a full proposal. And it is a very interactive process. Please, if you decide to write a proposal, feel free to reach out to our contacts to do that. That's the way we like to do business. The second type of proposal call supports collaborations between EMSL and JGI, has a rather fancy name of FICUS, which stands for Facilities Integrating Collaborations for User Science. I can never remember that, so you don't need to. But the cool thing about this for you is that you write one proposal and you get access to two facilities. Again, they're funded for 2 years. You are connected as the user, you're connected to capabilities at EMSL and at the Joint Genome Institute. And in addition to those, you also have additional capabilities available through our partners, including the National Ecological Observatory Network, which you may know as NEON, the Biosands Beamlight at HIFER through the Center for Structural Molecular Biology, and also the eBerLite program through the Advanced Photon Source. So the focus areas will come out when the full call opens on January 9 or thereabouts. Letters of intent will be due in mid-March, and full proposal will be due on May 4. All right, so the third of the 4 proposal types is another collaboration, in this case, another FICUS specifically. And in this case, it's to have joint access to capabilities at EMSL and at the Atmospheric Radiation Measurement Laboratory. So again, funded for 2 years, connects researchers with molecular characterization capabilities here at EMSL and sample collection capabilities at one of the ARM mobile sites deployed around the US, as well as science capabilities. Again, the specific focus area will be announced when the proposal call opens in just a few weeks on the 14th. Letters of intent are required if you want to submit a full proposal, due on February 9, full proposal due on March 23rd. And then the fourth type of call is actually not for a full proposal. It's actually the first of a brand new type of solicitation we're going to put out. It's called MONET Soil Sample Call. MONET stands for Molecular Observation Network. And in this case, there's 2 major things to understand. We're asking for the community to participate in this community science project to submit core samples and grab samples using a standardized procedure that we'll send you. And in return, you get a tremendous amount of information. We actually have a standardized workflow that produces 24 different data types, notably including high-resolution organic matter composition, the pore network structure, metagenomes, hydraulic properties, and a big long list of other biogeochemical parameters. This is free. So there's so many data types here that graduate students, postdocs, faculty who are running research programs that are at all related to soil ecology can get a big boost by just submitting a sample. We do it all. We do the analyses. We're trying to build up a database. It's a cutting-edge new database. The data will be publicly available upon deposition. And so you can send a sample, you get a ton of information, and it helps build up a molecular soil property database that will support new climate modeling and land atmosphere interaction modeling. So we'll initially release 2 topic areas. One will be soil function, and the other one will be rapid response, so that we can respond to science opportunities created by a short-term transient phenomena like flooding. We're hoping it will open on February 1st. This is a new type of solicitation. It's a little different from the other proposals. And in fact, exactly a month ago, we had a webinar on the Molecular Observation Network. And I encourage you to visit our webinar library where you'll see lots of other cool webinars there, but including the Monet one. So if you're interested, please go look at this. For more information, visit the EMSL website, and you can also contact us at emsl@pnnl.gov. And with that, I'll wrap up and pass it along to our next speaker, Swarup China, who will be talking about the new terrestrial ecosystem interfaces area. Sorry, terrestrial atmospheric processes. Sorry about that. I got distracted by the screen. I don't have control anymore, Jenna. Is that okay?
Yes, that is okay. Swarup, you should have control. And for we're seeing some chat notification about some of the links not being copyable. And just want to let you know that we are looking into it. And if we are not able to fix it before the end of the webinar, we will have them available to be emailed out to everyone.
Thank you, John, for the introduction. I'll talk about the relatively new integrated research platform that has been developed at EMSL, the terrestrial atmosphere processes, and what are the different research areas that we will cover under this topic, and highlight some instrumentation that you can use specific to answer some of the science questions that you have. So what this IRP studies is basically the cross coupling of land atmosphere feedback. So for example, if you have different emission from terrestrial source, either in the particle phase or different chemical species in the gas phase, and once they are emitted into the atmosphere, they have different chemical processes, and where they transform and produce a new secondary organic aerosols, or you can have a primary emission from plants and soils. And while they process in the boundary layer and eventually get to the cloud, where they can form a cloud condensation nuclei or can form as ice nucleating particles to create a cold cloud in the atmosphere. So this IRP specifically focus on these four major topic areas, which is the land atmosphere feedbacks, and really try to understand how the different molecular mechanism happening, or different processes, if we can able to explain those, and then the biosphere atmosphere interaction part, that is the emission of biogenic emission, either is from the gas phase species that forming the secondary organic aerosol, or in the primary phase, primary biological particles. And then the aerosol cloud interaction, when the particles get into the cloud and how they form in the cloud properties and the evolution of clouds. These topics are aligned with the Earth and Environmental System Science Strategic Science Plan, which state the impacts of land atmosphere interaction on aerosol and clouds. And these IRP focus on the generation of this data that can be used by the modelers to integrate into a model that has a larger impact of these different processes that we have. And these topics has been developed based on some of the previous workshop that we have, and also some of the breakout session that we have, different PI meetings. So now I'll briefly mention about different capabilities that we have to answer those four science topics that we have. So atmospheric aerosol in general is very complex, and you can see, for example, this is a electron microscopy image where we can see those different morphologies, and they can also have different chemical composition. And sometimes to understand different processes, you need to target different or multimodal approach to study these properties. And one thing you can see from the picture itself, this particles has a wide size range. So to do that, first thing we need to collect the sample that has a wide size range. So we have a new system that has been developed in collaboration with ARM, that is the size and time resolved aerosol collection systems. Once the particles are collected, you can look at using different microscopy techniques, either from a scanning electron microscopy or transmission electron microscopy, or if you want to look at the three-dimensional structure, you can look at that atomic force microscopy. You can look at the spectroscopy as well, looking at the chemical composition, or looking differently. For example, if you have a very small particles and you want to look at the chemical fingerprints, you can look at AFM Raman, or if you look at some correlated images from the SEM, and for example, nanosims can give you some isotopic weather information. as well as some time of flight SIMS measurements. And then we have the other approach we typically use is a mass spec, where you can use a different type of mass spectrometry approach. Again, I'm highlighting few that are commonly requested or used for the atmospheric science research here. So one is the nano-DC high resolution mass spec, which is very useful when you're looking at a small amount of samples, which is the case for atmospheric aerosols. And if you want to really look at the molecular composition great details, so you can use one of the FTICR instrument that can give you really the more complexity of different molecules that you have in your samples. We can also use a different multi-omics approach. This example typically showing a metabolomics, where looking at the small molecules here, and you can see how these atmospheric aerosols are diverse in terms of the different fingerprints they have. And other instrument is the mini spread, which is a very powerful instrument. Is a single particle mass spec, where you can look at the individual particle composition, size, shape, and volatility and evaporation kinetics to understand how the particles evolve in the atmosphere. And then we also have a ice nucleation chamber. This is a continuous flow diffusion chamber. So if you want to try to understand how the particles are nucleate ice, because only once in a million particle nucleate ice, so you want to understand and what are the different particles or what fraction of the particles nucleate ice. So you can use these ice nucleation chambers, for this particular talk, I will highlight only a few capabilities here that was recently used in some of our work. And again, if you want to go a little bit more details about some of the highlight, you can see here in our previous webinar about the aerosol characterization, and you can see all the details about the CCACM and ice nucleation chambers, as well as the mini spread instrument. So the first one I want to highlight about this van atmosphere feedback studies. If you want to look at the different processes that are happening in the boundary layer before reaching to the cloud, we try to understand how the size and time resolved chemistry happening in this atmosphere. So for this, with collaboration with ARM, we developed this size and time resolved aerosol correction system, which also has integrated with a different in situ sensors, for example, the size distribution measurement, as well as the black carbon concentration at different wavelengths. So what we do is bring those samples here at EMSL, and we can do a different kind of multimodal analysis using either microscopy, spectroscopy, or mass spec approach that was highlighted in the previous slides. Second example I want to highlight is that recent work, again in collaboration with the ARM, where we're looking at the molecular composition of organic aerosols in one of the ARM site at the southern Great Plain, where we collected aerosol at a different time during day and night, and try to understand the molecular composition. And what we observe, there's a lot of the CHNOS compound, which is typically the organosulfate you can find in the atmosphere. And typically this thing happen when you have a biogenic and anthropogenic interactions are happening. So for example, if you look at the wind dependence, like a plume coming from a south part of the town, where you can see a lot of anthropogenic emissions, and how these organosulfate are formed, how they differ day versus night, and how their volatility, for example, changes with during different time of the day. So something that you can understand. And again, this is something you can use one example again to how the molecular composition can help to understand some of this mixing mechanism in the atmosphere. And this whole article has been highlighted in a featured covered article in the journal. Next, this is my last slide, where I really want to kind of highlight different instrumentation that you can use for aerosol characterization. Again, this is just a subset of instrument list. You can go to the website for the full list. So first one is the sample collection. These are specifically for the balloon samples, where in collaboration with ARM, there is the size and time resolved aerosol collection system, or if you want to have a more amount or more bulk aerosol, you can use this time resolved bulk aerosol collection system. And then you're looking for atmospheric particle calculation, which you can do in two ways. One is the single particle approach. Another one is the bulk molecular composition. For single particle, you can look at the computer-controlled electron microscopy, where you can look at individual particles and get their chemical composition. You can, if you are interested looking into the internal structure, specifically of smaller particles, we typically look into the environmental transmission electron microscopy. And MiniSprat is an in situ instrument, as I mentioned before. So this is if you are interested looking at the, again, size without chemical composition, and either from a lab-based chamber studies or field measurement, something that you can request. WIPS is a bioaerosol sensor. This is something will be available starting from this fiscal year to the users, and also the analysis part of that. And also the AFM1, if you are very interested looking at the small particles as well as their chemical signatures. And for the bulk, if you are interested in the molecular composition, I think I already mentioned about the nano-DC high resolution mass spec. You can also integrate that into a FTIC instrument. And also, if you're specifically interested in the separation of specific compounds, you can do with the either liquid chromatography or gas chromatography, and that can be integrated into a mass analyzer. Next is the ice nucleating particle composition and the freezing mechanism. So one thing I did not mention is that there was an ice nucleation chamber as well in the environmental SEM that is integrated. And this is specifically useful for more fundamental studies where you really try to understand how individual particles freeze, especially the internally mixed particle, which portion of the particle is getting freezed, or if you really want to understand the thermodynamic properties, what relative humidity with respect to ice, or the temperature ice is formed. The ice nucleation chamber that was mentioned, this is more like a continuous diffusion chamber where you get more particle statistics. And then other one I want to mention is the MiniSprat, which you can integrate with the ice nucleation chamber. So you can separate out the ice crystal with an impactor and then get it into a MiniSprat. So you can get particles that nucleated, and you can also look at the particle population that, overall particle population. So you can get an idea which particle nucleated and which did not. And then you can also separate out those crystals and deposit on a substrate where you can look at the more like a morphology and the chemical composition, either with the different type of electron microscopy. So this is all I have. And so far we have been talking about how these atmosphere particles or gases, when they emit in the atmosphere, but when we look at the below ground, these processes also affect the emission of these particles. So that's what Amir was going to talk about in his next RP. And I forgot about this last one. Sorry about that. So this is a new instrument actually we have. This is a volatile organic compound measurements. Is offline measurement, a very sensitive instrument. So if you're very interested into looking at the VOCs emitted from plants or soil, so this is again also aligned with the next IRP that Amir going to talk about the below ground and how the plants are affecting. So if you're looking into the different emission from plants as well as soils, this is the instrument that you can use for the VOCs measurements. With that, I'll hand over to Amir. Amir, do you have access?
So not yet. So just trying.
I noticed there is a bit of a lag. Oh, someone's moving the laser pointer. Is that you, Amir?
No, I'm not using laser pointer yet. So, but I don't know why it doesn't move. Next. Oh, I think it works now, right?
Definitely a bit of a lag. Okay.
Okay. Okay, great. Thank you so much, Swarup. And hi, everyone. I'm Amir Ahkami. and by training, I'm a plant biologist, and currently I'm leading the integrated research platform on rhizosphere function, which is the newly developed IRP, which I'm going to talk about the science that we cover under this IRP, and also some of the capabilities and resources that associated with this IRP. Okay, so rhizosphere is usually defined as soil region influenced by plant roots, or sometimes even referred to just a thin or narrow zone of soil associated with plant roots. But beyond that, rhizosphere has some global implications that influence carbon sequestration and nutrient cycling, plant resilience and productivity, soil health, microbial structure and function. So this is a very complex system, and that comprise an integrated network of plant roots, soil, and diverse microbial consortium. So specifically in the rhizosphere, plant roots play a central role. So they take up water and nutrients and creating unique physical and biogeochemical environment for microbes, and emit atmospheric carbon into the soil. So rhizosphere significantly impact carbon flow and the transformation. So with all the complexity and various interacting components, there are many and remaining questions and exciting questions in this area and in the context of rhizosphere biology, which we try to cover under this IRP. But specifically, we are trying to understand questions around, how do root control processes influence carbon and nutrient biogeochemical cycles, and specifically developing of this new IRP for us is a new effort in re-scoping our vision in EMSL, mainly for us to be further in line with the Earth and Environmental Science Division of BER, and also to more specifically address, our user needs, user community needs in the science areas related to root microbe soil interactions. So just to give you a bit more of context here and highlight some of the major points, I should say that soil organic carbon formation is affected by obviously many soil factors, but that's not only that. So soil organic carbon formation is also affected largely by vegetation factors, such as plant biomass allocation among roots. So then rooting process and root structure by itself is the initial distribution of carbon in the soil by plants. The related important factor here is the rhizodeposition, which is defined as all material lost from plant roots, including water-soluble exudates. So root exudates actually acting as source of soil organic carbon and also prime microbial activity. As you see in this picture, soil holds twice as much carbon as does the atmosphere. And most soil carbon is derived from photosynthesis that takes carbon into root structure and then further into the soil through root exudation. And it's important to highlight this point that the ability to secrete a wide range of compounds into the rhizosphere is one of the most remarkable metabolic features of plant roots. According to some literatures, up to 20 or even 40% of total fixed carbon being transferred into the rhizosphere through root exudation. So another important factor to highlight here is that the carbon released in the rhizosphere leads to chemical, physical, and biological characteristics that differ from those of the bulk soil. So this is nicely shown in this study that I just highlight here, where hundreds of available amplicon sequences from the bulk soil and rhizosphere in different ecosystems around the world were used to kind of generalize bacterial characteristics regarding community diversity, composition, and function. The key findings were that the bacterial diversity in the rhizosphere was reduced and represents a subset of the bulk soil community. And for example, some specific genes, were specifically enriched in the rhizosphere, like the ones that involve in nitrogen fixation, but some of them were strongly depleted in the rhizosphere. So this overall suggests that the rhizosphere is the powerful factor shaping the composition, structure, and function of the soil microbiome, and therefore it is a key factor in the cycling of biogenic elements. So with that brief introduction, I just want to introduce this new IRP, that the key science areas that we're targeting under rhizosphere function IRP. So here we'll mainly investigate the molecular mechanisms of root-soil microinteractions and the effect of root control processes, including rhizodeposition on below-ground carbon flux, biochemical nutrient cycling, plant resilience, and microbial community structure and function. So we're specifically addressing the impact of root system architecture and root exudates on rhizosphere components in response to environmental perturbations. So now I want to highlight the fact that why we believe EMSL is a good place to do this type of studies, and what specific capabilities that we can offer to the user community to address questions in the context of rhizosphere biology. So being in the national lab and EMSL as a DO user facility enable us to getting access to state-of-the-art and integrated technologies to address research gaps in the area of rhizosphere biology. So being in EMSL actually uniquely position us to get access to these integrated capabilities. So for example, as you see in this picture, we have access to stable isotope probing or different type of imaging, root phenotyping. We have, different types of omics capabilities. In some cases, we can do spatially resolved omics. We have a phytotron that we can grow plants under controlled conditions, which there actually some of the chambers are equipped with carbon labeling chamber that enable us to do stable isotope probing. And of course, we also have some specific devices that built in-house, like synthetic soil habitats, which I'll talk a bit about those. So having access to those capabilities enable us to, follow these science areas and, investigating the flow of carbon and nutrients, discovery of signaling molecules, novel signaling molecules in the context of root-soil microbe interactions. Spatial and temporal patterns of root exudates can be investigated, and we can study the effect of that pattern in the nutrient cycling in the rhizosphere. And also, we can study the impact of plant biological diversity and root structure of the root exudation and microbial structure. And of course, we can study this process under environmental perturbations. Okay, so now I just want to highlight some of these capabilities in a bit more of details, although due to limited timing, I don't have time to really go to lots of details, but I think this just, give you, hopefully give you some good overview of what we can do in EMSL. And of course, I really encourage everyone to look at our website and reach out to us if you have any questions. So the first one is stable isotope probing or SIP. So we use this technology when we're trying to answer questions around the idea that how do plant species and microbial community regulate flows of carbon and nutrients, and how much of these processes vary over space and time. So we employ this approach to investigate the fate of carbon in the rhizosphere and to identify major regulators of carbon allocation toward roots and rhizosphere region by integrating this approach with other EMSL capabilities like multi-omics or chemical imaging. So for example, you can use IRMS here, isotope ratio mass spectrometry, to quantify the amount of the deposited isotope. And we also have a specific type of IRMS, laser ablation IRMS in EMSL for spatial determination of isotope deposition. So just to highlight one example here that how we use SIP technology very recently. So we studied sourcing carbon allocation process in Poplar, and we were able to employ SIP approach to monitor carbon transfer to root tissue under a given condition. And then using metabolomics, we identified a specific metabolite like sugar alcohols or TCA cycle compounds that could be linked to the higher carbon demands in the root system. So the other capability for us is to offer to users is X-ray computed tomography. So this is an advanced image-based technology for screening rhizosphere and below ground processes. So for this technology, plants are grown in pots, in soil, or in a preferred medium, and placed in an X-ray CT instrument for scanning. The plant in the pot rotates to generate a 3D volume of the root system, as you can see here. So I don't know why. Oh yeah, it works now. So as you see in this video, we can generate a 3D computer model of the root system in native soil, which is very informative for us to get access to specific root traits and root morphological and topological information. So XCT can be also hooked to omics platform and microbial analysis for decoding chemical language of plant communication in the rhizosphere. So for example, we integrate 3D root image cartography derived by X-ray computed tomography with metabolomics data to determine the spatial accumulation of metabolites in specific regions of root microbial hotspots. So more specifically, we use this device or 3D printed so-called rhizogrids, as you see in this picture. So you use this and put it in the pots as plants grown, develop the root system, and then you image them with X-ray computed tomography, generate that 3D model of the root system. And then, so this rhizogrid actually acts as a scaffold for physical support, and then you can get out the root system of the pot. And then from each quadrant of the rhizogrid layers, actually you can sample root tissue or soils around the root tissue, and then do microbial analysis or molecular profiling like metabolomics as we did here, and then map back those molecular and microbial information to the 3D root model. And then this way, we can get to know what specific metabolite or microbial hotspot are associated with a specific region of root structure. So the next is our synthetic soil habitat capability. So as you know, soil is spatially heterogeneous, and in soil, we cannot control all the variables to study details about biogeochemical processes. So that's why our scientists created these synthetic soil habitats. So with these systems, actually, when we use it, we can reduce the complexity of the root, of the soil, and then increase resolution so that we can manipulate parameters for testing hypotheses that we cannot test in real soil. Specifically, one type of this synthetic soil habitat, which is called rhizosphere on a chip, or you can even call it rhizochips here. So these are the platforms that mimic the physical structure of soil, as you see in this picture. And this is kind of a synthetic soil microhabitat that is compatible with plant growth and very helpful for rhizosphere studies. So, and you can see root system can navigate through the synthetic soil pore space. And it is the really good thing about this system is that it is compatible for tons of other capabilities that we have in EMSL, right? Optical microscopy or even chemical imaging. And as you see here, you see this in action when we grow poplar there, and you see poplar roots are navigating through these synthetic pores. and then we put it in the microscopy platform, and then we image it. So, for example, you can use fluorescently tagged microbes and inoculate in this system, and then monitor how those microbes can colonize, specific root cells in the root system. So my last slide is, on highlighting mass spectrometry imaging. So one critical need that enable us doing the research in the rhizosphere area is imaging complex molecular transformation at spatial scales within the root soil interface. So here in EMSL, our scientists develop different types of mass spec imaging, including MALDI. And as you see in this example, this is a section of the root system, and you can see accumulation of different metabolites, which colored in, you can see in different colors in each region of the root section, in each particular cell types, which is significantly important, for studying rhizosphere biology. And you can even use the same technology, in looking at the entire root system, and you see, the signals of each particular compound, an entire root system can be detected using this approach. More interestingly, we can also, apply this approach in studying, root soil and microbe interface in a controlled setup like rhizoboxes. So with that, we can, use some specific membrane and extract and, extract the compounds and transfer them to a specific membrane, and then image that membrane with MALDI approach, and then give us, the information of the active metabolic map in the rhizosphere region. So I don't want to talk about details on this because Dusan is the next speaker here, who is our expert in mass spec imaging. So he's going through all of these things in more details, and even describing a novel approach that he and his team is developing. So by that, I just want to thank you all, and please reach out by email if you have any questions, and there's a Q&A in the end. I'm happy to take questions. And by that, I just pass it to Dusan. Thank you.
Great. Can I control this? Yeah, okay, great. Thanks, Amir, for this really nice introduction. And my name is Dusan Velickovic, and today we will actually go over novel mass spectrometry imaging capability that we recently developed and implemented into EMSL user program for improved imaging and confident identification of primary and secondary metabolites that we can visualize in rhizosphere related samples. This is just a short overview of what you will learn today. Basically, I will give you some basics of mass spectrometry imaging, and then about the most commonly employed mass spectrometry imaging for spatial metabolomics and for molecular imaging, and it is MALDI mass spectrometry imaging. And then what are some general limitations of this approach, and why we had that need to improve it and to now offer it in the better version, and what you can actually see with this new capability that we developed. So mass spectrometry imaging is actually spatial probing or chemical probing of surface chemistry, of spatial surface chemistry. And what we do here, actually, we use probe that can be either laser-based or some kind of micro extraction. And we go from pixel to pixel, from area to area in the biological sample, acquire mass spectrum, and then that mass spectrum basically reflects molecular composition of that particular area that we probed. So when entire biological sample is scanned by mass spectrometer, we obtained ion images that basically reflect relative abundance of molecules of particular mass over biological sample. In this case, this is a root tip, for example. So this give you basically information about spatial distribution of molecules within biological samples. It's completely untargeted approach, so you can analyze hundreds to thousands molecules in the single run. In EMSL, we also have capabilities that can spatially analyze distribution of wide range of molecules, so from small metabolites up to the proteins. Today, I will focus mostly on the small molecules or metabolite imaging, and depending of also what spatial probe we use, spatial resolution of analysis can be from cell clusters to near single cell level. And what is also really important, and what I want to highlight, what put EMSL in front of other facilities that also has mass spectrometry imaging capabilities, are that EMSL is equipped with different additional tools that enable analysis of challenging environmental samples like rhizosphere samples are, and some of that already Amir hinted in his talk before. So the most common and most often employed molecular mass spectrometry imaging capability at EMSL is MALDI, or matrix-assisted laser desorption ionization. So that name comes because we need to use actually for spatial probing UV laser to ablate material and to ionize it. Because remember, in mass spectrometry, you need to ionize molecules if you want to measure them by the mass spectrometer. And then what is also important is that MALDI requires application of kind of coating of the with monolayer of MALDI matrix. Those compounds are usually small aromatic acids or bases that enhance and help in ionization of endogenous molecules. And in MALDI, our MALDI systems are coupled to two ultra-high mass resolving instruments that's called the FTICR. So we have a couple of them, 7 Tesla and 12 Tesla, and they provide really ultra-high mass resolution and mass accuracy. So we are really confident in assigning molecular formula to ion images that we detect. Also, MALDI sources that we use here provide you spatial resolution that is roughly 20 microns, and mass spectrometers have optimal analyzing range that is between 100 and 2,000 dem over Z. So putting it in the good range for metabolites and metabolomics, spatial metabolomics analysis. So Amir already hinted this, but I want to show you like just what kind of samples usually user send to us and what kind of questions we can answer with MALDI mass spectrometry imaging. So probably the most common employed sample types are roots or sometimes leaves as well. So of the plants that are in some kind of interaction with microbes or in some kind of abiotic or biotic stress. And then we can, for example, visualize metabolic translocation in those stresses or interaction with microbes. We can also create a 3D metabolic map of entire plant tissues, like in this case, for example, soybean root nodule. We can also, as Amir said, visualize hotspots directly in the rhizosphere. We can also analyze microbial systems, and we can provide metabolic topography of microbial colonies, and then do like depth profiling, and based on that, decipher different phenotypes in the biofilms. And finally, also one of the most common samples that we analyze here are solid agar plates where different microbes are interacting, and then we can basically image metabolic exchange on that microbial interface. However, MALDI mass spectrometry imaging and mass spectrometry imaging in general has some limitations. Two most common are that many important molecules still remain undetected. That is especially true for low abundant molecules, and also for poorly ionizable molecules, because remember, molecules need to be ionized if you want to see them in the mass spectrometer. And then also molecules that are below 100 Daltons, they are out of range of our instrument, of optimal range of measurement of our instrument. So that is also a problem, and a lot of phytocompounds, for example, pyruvic acid and some other aldehydes, are also non-detectable in traditional MALDI mass spectrometry imaging workflow. And the second challenge is actually confidence in molecular identification. Metabolites are small molecules, and because of that, they have like low amount of carbon, low number of carbon atoms. And because of that, especially for the low abundant molecules, C13 isotopes are very often below limit of detections. And because of that, because we use for molecular identification and for assigning accurate molecular formula, we use fine isotopic pattern, we are basically limited here because we cannot see anything like those higher isotopes like C13 and etc. Also, problem that is inherent to the mass spectrometry is that species that are isomeric, actually, that have the exact same molecular formula, even if they have different functional group, are undistinguishable in MALDI mass spectrometry imaging. It is reason why I cannot move the slide though. And I don't move this cursor now. Oh yeah, that's good. Thanks. So it is reason why our team recently developed on tissue derivatization workflow that can bypass those challenges. So at the first place, we basically spray over the tissue sample derivatization agent called 4-EPIBA. And this derivatization agent actually will, because it will attach to the analytes, will push those metabolites toward higher M/Z that is optimal for mass spectrometer measurements. But also what is important, it will introduce permanent charge to the analyte. So it will be in form of the ion, and in that sense, sensitivity will be increased. But also what is important, it will introduce bromine atom into the analyte, which you will see on the next slide, is important for more confident annotation of molecules that we see. Also, what is important is that this workflow that we developed enable different derivatization of carbonyls, of different carbonyls. So even if we have like mass isomers that have different carbonyls there, like carboxyl groups, ketones or aldehyde, by using our workflow, we are able to differentiate them actually. Next slide. I cannot move for some reason. Yeah. Sorry for that, but I cannot just move my slides. Okay. Oh, okay, thank you. This is example how this derivatization workflow actually improved confidence in metabolite annotation, because we, as we introduce bromine atom, we actually introduce a unique isotopic pattern, because bromine has two isotopes, 79 and 81, that has relatively same abundance. And in that case, it's very easy for us to do theoretical with experimental matching of isotopic patterns and to confidently annotate the metabolites. This is on example, for example, on abscisic acid, so really low abundant phytocompound, where we were able to actually detect it in specific cells in the poplar root cross-section. But also because we increase sensitivity of the analysis, we were able also to image other phytocompounds like jasmonic acid, the methyl jasmonate, salicylic acid, also growth hormone, aminocyclopropan carboxylate. What I also want, can you go on the next slide? Because, yeah, thank you. Also, what I want to highlight here, that because we increased sensitivity of our analysis, our new derivatization workflow enable us to basically visualize the entire TCA cycle in plants, or also to visualize different other biologically important carbonyls, like very low molecular mass acetaldehyde, but also reducing sugars, malonic acid, etc. And my last slide, if you can move on the next slide. Yeah, and this is last slide. What I want to highlight here is that MALDI mass spectrometry imaging is compatible, and this new derivatization capability is compatible with other sample times offered to EMSL users. And Amir talked about those rhizochips. And here you can see example how by using MALDI mass spectrometry imaging, we can actually see and detect metabolites that are present in the root hairs, and that basically exude into the root pore, into the soil pores. But also we can detect other types of molecules that are present only in the tissue of the root. With that, I would like, to conclude. So, and I think we are open for questions, I guess. Thank you. And sorry for, something went wrong.
You did great. Thank you. All right. We do have a few questions, and I know that we're kind of running out of time, so I'm going to toss a few questions to our speakers, and then if we don't get to everything, just know that we will reach out via email with some answers. The first question is from Ann Summers. It's for Dusan. Does MALDI pick up soil inorganics, both nutritional and toxics?
So not inorganics though. So we are limited really on the molecular, like those are usually like metabolites that are, I would say, like above like